In this video, we're going to look at the structure of DNA. And DNA, as you probably are aware, is one of the most, or if not the most important molecule in all of biology, as DNA is responsible for making each individual and each different species and living thing unique and different on the planet. Many species share a lot of their DNA code or sequence in common, but small differences in different genes and different amounts of DNA can result in some big difference in physical or behavioral or physiological traits. And so in this video, we're going to look at just the structure of DNA and how that structure was actually discovered. And this is IB sections 3.3 and 7.1 for the 2016 starting exam curriculum. So to start out, DNA is made of small pieces, building blocks you could call them, called nucleotides. And nucleotides are composed of three different parts: a phosphate group, a sugar, and a nitrogen base. And there's four different types of nitrogen bases: adenine, guanine, thymine, and cytosine. And so when you see DNA, you probably oftentimes have seen it written just as single individual letters. And the reason for that is each one of those letters, A, G, T, or C, stands for one of these nitrogen bases, types of nitrogen bases. Each nucleotide has a phosphate group and a sugar, and that sugar is deoxyribose. And all nucleotides in DNA have a phosphate group, a deoxyribose sugar, and then one of those four nitrogen bases. And so what makes what makes the sequence of DNA different is the order or the sequence of the different types of nitrogen bases that are in those nucleotides. Those nitrogen bases in these nucleotides can be grouped into two different categories. One is called the pyrimidines, and the second is called the purines. Thymine and cytosine make up the pyrimidines, and adenine and guanine make up the purines. When DNA is matching up, DNA is a double-stranded or a double helix, a pyrimidine matches with a purine. And what happens is the thymine matches with the adenine and cytosine matches with guanine. And the reason for this is that they form different numbers of hydrogen bonds in order to connect the two strands of DNA. And we'll take a little bit more, closer look at that in class and a little bit later in the notes video as well. Now, if we're looking at DNA, we also have to consider RNA, as RNA is also a type of nucleic acid, and there's some minor differences between them. The first is in the type of sugar. DNA is deoxyribose. And ribose sugar is used in RNA. In RNA, the thymine is replaced by a molecule called uracil. And one of the other large differences is that a single strand is only present in RNA, whereas a double strand is present in DNA. And so those are the primary differences: single strand, thymine and uracil, and then the type of sugar. Also, DNA is only ever found in the nucleus, unless the cell is undergoing mitosis. Otherwise, RNA can be found both inside the nucleus as well as out in the cytoplasm during transcription and translation. Now, how is DNA held together? Well, there's a couple different types of bonds that help to hold DNA together. And in looking at specifically the connection of different nucleotides, so here's the phosphate, here's the sugar, ribose, in this case this would be RNA, and then our nitrogen base. The sugar is always attached to the nitrogen base, or I should say the base is always attached to the sugar, which is connected to the phosphate. And so if you want to say that there's a backbone to the structure of DNA, the backbone is composed of phosphate and sugar. And it is in a repeating order: phosphate, sugar, phosphate, sugar. So here we can see that phosphate and the sugar, phosphate and the sugar. The bond that occurs between the two nucleotides, between the phosphate and the sugar, is called a phosphodiester bond. And so in order to build a polymer of nucleic acids, nucleotides are put together. And DNA and RNA are polymers of nucleic acids. It's just multiple of these nucleotides put together in a specific sequence. In looking at the structure of DNA more specifically and overall, we can see here's our phosphate, here's the sugar, phosphate, sugar. This is our backbone right here. In the middle, and attached to each sugar, is the nitrogen base, a particular type of nitrogen base. And so thymine here is matched with adenine, and between those two there are two hydrogen bonds. Between guanine and cytosine there are three hydrogen bonds. And this is important because it helps to ensure that, or was one of the reasons why thymine is always going to match with adenine, and guanine is always going to match with cytosine. It is possible for a mismatch to occur, and that would be a type of mutation. Sometimes that's possible, not always, but sometimes that is possible. One of the other important things to mention and point out in this diagram is the anti-parallel arrangement. And so what you can see is that the phosphates are pointed in opposite directions, and they're also labeled, each end of this molecule is labeled with a 3' or a 5'. And what this means is that the direction of the molecule is flip-flopped or is anti-parallel to one another. And so in this situation, on this chain here, the direction is 5' is on the top, whereas on the opposite 3' is on the top. And this is going to become really important when we get to DNA replication and get into those more specifically. So to recap, hydrogen bonds are holding the two strands together. So this is a strand and this is a strand. And those two strands are held together by hydrogen bonds in between the different nitrogen bases. Phosphodiester bonds hold together the individual nucleotides. So here's a phosphodiester bond between this phosphate and this deoxyribose sugar. And that's what helps hold these nucleotides together in order to make DNA and RNA. So going back to the anti-parallel arrangement, each strand of DNA is arranged in the opposite direction. In relation to one another, they both have 3' and 5' ends. One end of the nucleotide has a phosphate attached to the 5' carbon, and one end has a hydroxyl group attached to the 3' carbon. And then nucleotides are held together by phosphodiester bonds. The double helix shape and the holding of DNA strands is caused by hydrogen bonds, to recap. Now the structure of DNA goes back to a number of different scientists who are responsible for it. Watson and Crick are two scientists that you've probably heard of before, and they are the ones that are generally thought of and given most credit for identifying the double helix structure of DNA. But there are a number of other scientists involved in the discovery of DNA, and one of those in particular was Rosalind Franklin. Rosalind Franklin spent time in the lab and was actually able to produce the first photograph of DNA. This is called Photo 51, and it's an X-ray diffraction, and it shows this X pattern, and this provided evidence for the double helix through this photo here. And unfortunately, actually through her work, Franklin ended up developing cancer and eventually dying because of the radiation that was caused by producing these X-ray diffraction photos. But her research and the discoveries that she made, in particular this photo of DNA, helped to provide the evidence that Watson and Crick then used to build and test their model of a double helix. The first discovery was the discovery of anti-parallel strands, and then Watson and Crick went on to hypothesize that there was actually a triple helix, and later, based off of continued data and experiments performed, identified that there were hydrogen bonds between adenine and thymine and guanine and cytosine between those nitrogen bases, and developed the double helix model. And so the discovery and the collection of evidence, particularly of Rosalind Franklin's photos of X-ray diffraction, helped to provide evidence that was necessary for Watson and Crick to be able to determine the overall shape and structure of DNA. Additionally, this model also suggested a means of replication, and if there's an anti-parallel arrangement of DNA, it suggested a means of how that DNA molecule is actually replicated, and that is what we'll get into in our next video, DNA replication.